Our next speaker is Dan Murphy. Dan arrived in Australia in 1999 after spending 4 years conducting agriculture research in the UK. He currently manages a research program that addresses issues relating to the development of sustainable management practices for agriculture, horticulture and mine sites under rehabilitation. His research is primarily funded through the Australian Grains Research and Development Corporation, the Department of Agriculture, Forestry, the Fisheries and Forestries, the Australian Research Council and various industry partners. Dan's key areas of research are microbial nutrient cycling, microbial ecology, soil biological fertility and carbon sequestration farming systems. Dan.
Thanks very much for the introduction. And can you hear me up at the back? Excellent, thanks. So I have been asked to speak about soil organic matter and microbial biomass. What I mean by microbial biomass is the mass of microorganisms in the soil. So Sandy's done a really good introduction to my talk because he pointed out the importance of key groups, rhizobia and mycorrhiza. They're individual types of organisms. Think of the microbial biomass as the total mass, as a weight of organisms in soil, and we use that as an indicator of soil fertility. And on the front slide I've put a tractor, because at the end of the day, what we're really interested with soil organic matter and microbial biomass is how we manage the system. What are we doing to the soil when we manage it for agricultural production? How does that affect our carbon storage? How does that affect our microbial populations? So we've been involved in two programs. One is, you've seen this map before, one is the DAF carbon program that was also supported by GRDC. So I'm from the west, so we've got all these lovely sandy soils, and we actually classify them into about 12 different types of sands, just so we think we've got a bit of difference in there. So it keeps us happy when we look at all your soils on the left. And what we've been doing is collecting carbon stocks, but also trying to understand the fundamentals of how we actually change carbon stores in soil, because we want to be able to manage that to actually build our carbon, but also stay economically profitable. And the other program that I'll just mention briefly, because Andrew Werrett's going to give a talk on this after me, is the soil quality program, which has been funded through a lot of different funding bodies, but currently through the grains industry. They have a soil biology initiative. And this is really about looking at soil quality on a national platform and really trying to understand what are our constraints to production in an area? What's our constraints to net primary productivity? And the reason I mention these two programs is the DAF carbon program data sets nationally and the GRDC soil quality data sets that have been collected have often been done on the same soil, so there's been a co-sharing of soil samples, but all of that data is also on a website that Andrew will mention to you later. So it's a freely accessible way of actually looking at data from your area and understanding what some of the key issues are. The way I think about soil quality is very much, I guess, from a production point of view, with the idea that we've got plant growth in here. So unless you're irrigating, that's a water limited environment, we can grow a certain amount of plant material, and that's starting to define how much organic carbon, how much microbial population we can support in that soil. And we know that climate's a very strong driver, and Mick showed you a very good relationship for this area between rainfall and carbon stocks. We know there's a lot of things in the soil that affect biology, a lot of things in the soil that affect carbon store, because of course the microbes, the soil microbial biomass, they're eating that organic matter as their food source. I've just had a scone, so I'm standing here and my body is breaking down that scone, generating energy, generating heat, that's how we live, and we're respiring out CO2, carbon, CO2. Microbes are doing the same thing. They're taking that piece of plant material, they're breaking it down to generate energy, and they're releasing CO2 back to the atmosphere. So think about plant growth driving biological processes, think about how management alters that plant growth, and think about how subsoil or topsoil issues within the soil are affecting it. So I think Lachlan mentioned aluminium. If you've got an aluminium constraint, you're going to grow less plant biomass. Maybe you need to look at liming as an example to alleviate, by increasing pH, alleviate that aluminium issue, allow more plant biomass. So really, this is our tractor, we're thinking about how we fix our management of those problems. And the reason I circle the biology is I think that when we started this program, and what other groups have been finding as well that run workshops, is often the biology that we know least about. But it's central to our existence. Biochemical processes, chemical transformations in soil, biology at the front of it, biologically mediated transformations are driven by microbial populations. We need them to sustain life, we need them to grow plants. Soils are teeming with life. They're very, very high population numbers. So we're talking here about more bacteria on Earth than stars in the known universe. More stars in the known universe than there are sand grains on our beaches. Very, very high population numbers, colonising the soil. Interestingly, they colonise about 1% of the surface area of the soil. So you have very, very high population numbers surviving in niches, in microsites in the soil where the habitat is suitable. Microbes are a living organism. They're looking for a place where there's exchange of gases, oxygen, water availability, they can get rid of pollutants, they can find that food source. Big numbers, very, very high population numbers, very small surface area of the soil being colonised. And if you even think down to people like us, humans, if an alien came and digested us down in a chemistry lab and analysed us, they would think that we were bacteria, because we have a lot more bacteria than we do human cells. So there's a very tight interaction in our life with bacteria, and same if you think about the living soil, there's a very tight interaction with microbes. Why do we focus on the bit that's hard to see? Why do we focus on the bacteria, things like rhizobia that Sandy mentioned, the fungi, the mycorrhiza? Well, actually, although they're the small parts, they're the powerhouse, they're the energy source of the soil. And if we look here, a wheat example or a pasture example, and we look at the bacteria and fungi, the microbial biomass, the microorganisms in soil, or we compare them to fauna, larger organisms, earthworms, ants and so forth, most of the organic matter turnover in soil is by that small-scale biology. Larger organisms fragment, they break up organic matter, they break up plant material, but it's that bacteria and fungi that are doing the biochemical cycling that we need to re-release nutrients that become plant available, to re-release carbon, to allow that biological cycling to occur. So we talk to our farmers, we run workshops, and we ask them what's important to them and why they think soil organic carbon is important. And a key point that we recognise is nutrient supply. Microbes break down those plant residues so they don't keep on accumulating, and they release nutrients. It's a key part of how living systems cycle. Unfortunately for carbon storage, microbes are breaking down the organic matter, releasing nutrients that are plant available, but in doing so, they're actually making it harder for us to store carbon. So we have this tipping scale in soils between trying to build carbon by growing more plant material, by removing constraints to production, by fixing our agricultural management, but at the same time we have microbes trying to break it down. Organic matter is central to a number of functions, which is why it's such a key indicator and one of the primary things that we measure in soil. And here are just some examples. Energy for microbes, retaining nutrients, because of course we talk about soil carbon, but in that organic matter we have nitrogen, phosphorus and sulphur and so forth. Buffering pH in my sandy soils where we don't have a lot of clay, organic matter is central to buffering of nutrients. Physical, storing water, improving infiltration, getting that water into the soil that we heard this morning when Michael spoke. We measure soil organic matter, but it's not all the same. If you think about it, some of that soil organic matter has been sitting in that soil pre-European settlement of Australia, pre-agricultural management of soils, and some of it's fallen a minute ago when that leaf has just died or that root cell has just broken. So we have a continuum. Researchers like to break it up into different pools or fractions because it makes it easy for us to measure, but it's really a continuum from a bit of sugar, the icing on the cake, right through to a bit of tree lignin that's really hard to break down. They don't behave the same in soil. Different bits of organic matter behave differently. And if we think about it, and these are the turnover times or the decay times, a little bit of terminology here. So we're talking about particulate organic carbon, the more labile fraction, fresh residues breaking down, the very large humus pool, that's a very large nutrient store, or the more stable or recalcitrant or inert type of organic matter. Different turnover times, some within years, your legume residue you see disappear in a few years, a tree trunk sits there for generations. When you start examining specific components of that organic matter, you can get very clear relationships to biology and through to nutrient cycling and therefore through to plant capture. So here's an example where on the x-axis we've measured the mass of microbes, the weight of the microbes, their carbon content, and on the y-axis we've measured the nitrogen supply from that soil. This is the rate at which the soil is releasing nitrogen that is plant available. And you can see a very strong correlation as we get a higher population of microbes, more biological cycling, more nutrient release from that organic matter. We now focus on this little schematic up here. 66% there means that in this analysis of data, this is from the soil quality website that Andrew will show you in a minute, we can explain 2/3 of the variability in microbial biomass based on 3 measurements: clay content, pH, and that particulate organic matter, the bit of the organic matter that is highly microbially available. Why clay content? Well, if you think about a soil, as a soil gets aggregation and gets structure, it's building more homes, it's a habitat, it's where the microbes are living. So think of clay as the structure of the soil, the physics of the soil, the habitat for the microbes. pH? Well, all living organisms, including us, have a pH range that we live in. Microbes tend to prefer pHs that are more neutral. And particulate organic matter, it's their food source. If you're providing more food, if you've got a farming system that's putting more organic matter back into that soil, you're feeding that microbial population. Slightly more complex figure, trying to illustrate the same point here about why organic matter is important and why we now start to look at different components of the organic matter. The way you interpret this slide is, this is increasing clay, so I'm down here sitting in, a lot of our soils are at 2% clay, very, very sandy, right up here to, very, very hard to manage clay soils. If we look at cation exchange capacity, a very standard measurement that people get done, a measure of the capacity of the soil to retain nutrients. Cation exchange capacity is generated by clay content or by organic matter. So in my sandy soils down here, I have a very large influence from organic matter, which is why this is so wide, a very large influence from organic matter on cation exchange capacity. But not all organic matter, specifically the humus and the inert. So the really old organic matter that's sitting in the soil is driving cation exchange capacity, not the new stuff that we're putting in. Energy for biological processes, important regardless of what clay content of the soil it is. And you can see here this particulate fraction is driving a lot of the energy, where the inert stuff isn't, they're not breaking it down. So what we're starting to find is you can have 2 soils with exactly the same total carbon value, but they behave very differently in terms of biological functioning because they vary in their ratios between particulate, humus and inert organic matter. So that complicates the story a little bit in terms of interpreting how your management drives biological processes. So thinking about what factors most influence it, because that's what we're trying to do, we're trying to change carbon stocks. Data from our farmers, when we spoke to them, were again spot on, recognising the importance of climate and soil type. Climate, how much plant material can you grow? Soil type, how much plant material can you grow? But also what microbial populations you can hold. So thinking about climate, what I think is interesting, what I think is an important message, is when we started thinking about the carbon program a couple of years ago, 2009, most people were saying we should really focus our activities on the high rainfall areas because we've got more rainfall, therefore we can grow more plant material. Nothing wrong with that logic, except for what we find, and this is data from WA, running from 400 millimetres inland to about 750 on the coast. We actually find that where we have higher rainfall, our systems, happen to be annual pastures or perennials, are actually a lot closer to what we think our attainable carbon value is. Our attainable carbon value, or our upper limit, we have generated by modelling. There's a very good carbon model that has been validated for Australian conditions that we use. And what you're seeing is that as we increase in rainfall, we're actually finding that our measured values, these are all different farm paddocks, are actually closer to, or in some cases, actually above what we think is possible. Probably where water is moving in and they're getting underfed water that we're not accounting for. And really, it's our cropping systems down here that have the larger capacity to sequester more carbon. Lower rainfall, they're going to produce less biomass, but their capacity is larger. So we've got to think a little bit about that. Do we run for more rainfall, produce more plant material, or do we try to identify regions in our landscape, regions in our catchments, where we think the difference between what we're measuring today and what is possible, and go for the regions, we believe the right approach is to go for the regions where there's more capacity left in that soil to store carbon. And even in our sandy soils, these are gutless soils, we're still talking here about potentially having about 50 tonnes of extra carbon storage based on our rainfall and based on our sandy soils. Thanks. Why do you have to put more organic matter into sandy soils to build carbon than clay soils? This is a figure from Peter Grace, who works up in Queensland at one of the universities, and he's showing cation exchange capacity, but if you don't think in terms of cation exchange capacity, you can also correlate this to clay content. So as cation exchange capacity increases, the clay content of your soil is increasing, the physical capacity to house microbes but to sequester carbon increases. And this line here is telling you for any cation exchange capacity what the percentage of input is that's retained. So in my sandy soils, 0 to 5 cation exchange capacity, we retain less than 25% of the carbon that it goes in as organic matter. 25% is retained, 75% is going off as carbon dioxide. In our sandy soils, what is happening is the microbes can always colonise that organic matter because it's not getting physically protected by clay. If you've got a clay platelet sticking on top of your organic matter, it's going to stop microbial breakdown. Microbes don't eat through clay. If it's in a sandy soil where the organic matter is exposed, it's very easy for them to break it down. So there's definitely a soil textural reason why it's easy to store carbon in heavier soils. And likewise, microbial populations. So we would expect higher microbial populations in heavier soils than in sandy soils. And again, remembering that clay content is one of the key drivers for our habitat. So when we look at data on the soil quality website, we know that texture of a soil is going to change the values that we would expect to measure. So best practice, what can we do to try to improve the system while still staying economically viable? Well, I think it was quite interesting that when we ask our farmers, they're very focused on stubble management. So I want to really give a couple of illustrations about that. And it was interesting to hear Mick's talk where he was saying how through the management practices that were tested here, how hard it was to show differences in carbon storage. I want to try to maybe explain why we don't often see the differences as large as we would expect. So here's some data from a trial in WA that has been running for 18 years. After 18 years, very simple trial, either burnt or retained, same carbon value. Retained system has more particulate organic matter or labile carbon, the stuff that microbes can eat, and it has a larger microbial population, 50% larger. Two soils, same carbon value, different biological functioning. The straw retained system has a far higher level of biological cycling, and that actually translates in this trial through to additional nitrogen supply to the crop, and that translates through to a grain protein increase. But why might we think that after 18 years, we've been burning all the straw, we haven't built up any more carbon? Let's think about that. Below ground, our root systems are the same. if we're burning the stubble on the surface or we're retaining the stubble, our carbon input from our root systems are the same. In a wheat crop, about half of our carbon is below ground in the roots, so we're not changing that. Above ground, when it's burnt, we're putting a match to it, so the carbon goes up as carbon dioxide when we burn. In the stubble retained system, the microbes are still breaking down that organic matter, they're still breaking down that stubble as an energy source and still releasing 75% of that carbon as carbon dioxide. Our burning efficiency is probably no more than 75%. So really what we're doing is we're not changing our total carbon balance whether we're burning or retaining. What we're doing is we're changing whether we allow the biology to cycle that and allow for greater nutrient cycling in the soil or whether we're putting a match to it. But it's very difficult with management practices to sometimes change carbon because the biology is still breaking down that organic matter. And to finish off with, I just want to give another illustration where we ran a trial that's been going now for 10 years with the Levy Group, one of the large farming groups in WA. And what we did at this trial is we looked at carbon stocks under a modelled maximum value. So this red line up here is... So the red line up here is what we think is attainable. So none of the treatments after 10 years have got there yet to their maximum. It takes time to change carbon. It's a decadal time frame. You've got to wait a while. Just focus on this one here where we've tilled the soil, aerate in the soil. Microbes can break down the organic matter. Load up organic matter, tilled plus organic matter on this end, is where they've imported in hay, and over an 8-year period they added an extra 60 tonnes of hay to this treatment. And that extra 60 tonnes of hay resulted in an 8-tonne change in soil carbon. So for putting on 60 we've sequestered 8. To do the maths on that, how come 60 tonnes converted through to 8 tonnes of soil carbon? Well, of that 60 tonnes, plant material is about 45% carbon because it's made up of water and nitrogen and oxygen and phosphorus and sulphur and all the other things that goes into making a plant. So out of 60 tonnes, we end up with 27 tonnes of carbon going into the system. For this clay content of this soil, 30% is retained and the microbes respire off the other 70%. So really, 27 tonnes of carbon going in, an efficiency of 30% being retained, means that that soil can hold, or should hold, 8.1 tonnes, and we measured 8. So the maths work. You've got to put a lot of organic matter into the soil to sequester a fraction of that as soil carbon. This figure is in your book, so I'll leave it with you, but what it illustrates is that whole concept of we have a potential up here, a mathematical potential which is very much driven by soil type. That's our attainable value in our soil that we can model. We have climatic drivers, we have our actual soil carbon contents which are typically less than what we can store, and how we change our management is going to influence whether that actual carbon value goes up or down, but it certainly won't go any higher than what our potential cap is for our soil type. So I think some take-home messages. It's all about how much new biomass we can grow. Are we driving our system for maximal plant growth? How much of that plant growth do we export in animals or in grain is going to affect how much soil carbon we can build. You've got to do something profitable. Don't start only farming for carbon. Farm to stay in the business. Think about the economics, and hopefully by improving your systems, you'll actually start increasing your soil carbon, or at least maintaining them, and have other benefits such as nutrient cycling. And in terms of soil health, in terms of thinking about the soil biology, again, soil carbon is the powerhouse, it's the food source. So we're feeding our microbes and we're keeping that biological system running. But again, you have to do it in a profitable way. There's no point farming a system for keeping the biology happy if you're not making money out of the system. Thank you very much.